Okay, welcome everyone to our webinar. We'll spend some time and introduce the topic in a second. We're just waiting for some more people to join us, and then we will get started. This webinar is being recorded for future use, so if you need anything from it in the future, you can just feel free to let us know. We're supposed to have upwards of around 40 people in attendance, scheduled for an hour, so we'll get started. If you have any questions, there's a chat feature at the bottom of your window, Zoom window screen here, and you can enter questions. I can't hear you speaking, but if you put that chat, I'll try to answer questions at the end as best I can. So let's get started. Okay. So the topic of the webinar is attempting to understand and train brain networks. My name is Robert Coben. For those of you who don't know me, I'm a neuropsychologist. I've been in the field of EEG, QEEG, and neurofeedback for almost 20 years now, and I've specialized in connectivity analyses and treatment in autism and several other conditions. So this will be part one of the webinar, which will focus mainly on understanding brain networks and how we can potentially assess functioning in those networks. And part two will focus more on the application of that and intervention strategies as well. But today we'll focus on part one. As you can see from your initial screen, we'll deal with 6 sort of general categories of topics, going from different types of connectivity that can be measured to comparing these approaches, to a brief introduction to some actual structural brain pathways, then to an understanding of graph theory analysis, which will lead us into functional brain networks. And then can we actually measure effective connectivity in real life in our everyday practice and patients? This webinar enables you for BCIA and APA CEUs. And if you have any questions about that, feel free to contact us or Susan, our administrator, anytime after the webinar. The accuracy, utility, and validity of the information presented, to the best of my knowledge, will be very good, I hope. The majority of this information, I should say all of this information we'll talk about today, is taken from empirical scientific literature and our enhanced understanding of this information. There is no commercial support provided for this webinar, other than I am one of the directors of Integrated Neuroscience Services that does provide assessment and consultation services to clinicians. So there is that to report to all of you, and you can appreciate what we will talk about in that context. So we'll move on from there. In his landmark text published in 2009, Olaf Sporns delineated 3 different types of connectivity that could be studied in the brain: structural, functional, and effective connectivity. And we're going to run through each to help you understand what they each represent. Structural connectivity is a set of physical or anatomical connections that actually link neural elements in the brain. So this is the structure, the white matter tracks in the brain that can, and the neurons and neuronal pathways that actually carry information. So in our world, this is the real thing. Diffusion tensor imaging done from an MRI scanner is the only known technique that can measure structural connectivity in a live human being. It's been in the field now for about 10 years, and it actually, in an MRI machine, measures the transfer of water molecules from one location to another and actually does measure structural connectivity. This is what we are hoping to emulate when we look at other measures of connectivity if we don't have access to a diffusion tensor study in a given patient, which of course most of us don't. Functional connectivity reflects patterns of deviation from statistical independence between distributed and spatially remote neuronal units. Well, what does that mean? That means these are patterns of similarity or differences between regions that are separate from each other, usually taken between two regions at a time, but we'll talk more about that. The basis of this is time series data from neural recordings. So the classic example is a coherence measurement. We'll get into the definition in a few moments, but it's basically judging the correlation between two signals at any given point in time, regardless of what technique you use, if it's fMRI, PET, EEG, SPECT, MEG, etc. Their relation is taken as neuronal coupling and often takes the form of correlation, coherence, phase locking, or co-modulation. By definition, there is no causal relationship, effect, or interaction. So essentially, we're talking about correlational techniques that measure similarity in activity between two or more different locations, often two locations at a time. And correlation just tells you how similar they are. It does not tell you that one is causing the other, that they're causing it in any particular direction. They just tell you that they are similar at a given point in time. As such, we cannot infer causation, interaction, or directionality in any way. So in functional MRI, where someone's in an MRI machine asked to perform a certain activity, and their oxygenation of their brain is measured during this activity. What's called the bold signal in the fMRI, which is a heightened signal that detects oxygenation, is taken to reflect activity in those regions of the brain in a response to the stimuli that someone is focused on. We can measure similarity between two signals. For example, the posterior cingulate cortex in the parietal lobes and the prefrontal cortex. And if they are similar, we infer that they have functional connectivity. Okay, so in the lines you see on the bottom, you can see how the paths go up and down, and to the degree they're correlated at any one given point in time, we say that they are connected functionally. That does not necessarily mean that they're connected structurally or that they are causing each other in any way. Remember, this is functional connectivity, not structural connectivity. We can also infer this from PET scan or positron emission tomography, where locations, say, over the frontal lobes on the left and right side, if they have similar colors in these graphs, they're said to be doing similar functions, doing similar things at similar times. And we then infer that there is a degree of functional connectivity as a result. Of course, what we work with, or at least most of us in everyday basis, is EEG similarity. Of course, this image is of more than 19 channels of EEG data, but it works the same way. Generally, our coherence or phase measures take two locations on the surface of the brain, and they measure the similarity between those two locations at a given point in time, oftentimes within a given frequency in the EEG, theta, alpha, beta, or what have you. And we determine this value between these two locations. Again, this is functional connectivity or a correlational statistic between those two locations only, and we don't know what's happening at other locations or how activity at other regions may affect our locations of interest. That takes us to what we call effective connectivity. Effective connectivity implies network and causal effects between neural elements. It is inferred through statistical techniques such as time series analyses and statistical modeling that assess causality and interaction. So now we are no longer in the correlational domain where we can only establish that there's a relationship and not the cause. Now we are in multivariate autoregressive models and techniques that actually measure cause and the interaction of causes between different locations. This requires complex data processing and modeling techniques such as independent components analysis, which is what ICA stands for, partial directed coherence, and something called Granger Causality, which enables us to measure causal relations between locations, either at the surface or the source of the EEG activity, how one region causes another, and how they mutually and reciprocally cause each other. So we can measure interactions back and forth, the directionality, and the causal and interactive components of this level of connectivity. In this model of connectivity, we don't just see two locations and correlate them. As you can see in these images, you'll see between the locations there are actually arrows with directionality to them. So we can measure how point A causes the activity at point B. There is no longer any dispute or any uncertainty about whether one location causes another, as is true in correlational or coherence techniques. Now there is direct cause and regression of that information from one site to another and back in return from A to B and B to A, so we can measure reciprocal causality as well. It is generally considered that measures of effective connectivity most resemble actual structural connectivity. That is the idea behind developing effective connectivity metrics, whereas measures of functional connectivity do not reflect this, and we should not expect this to be the case. This, of course, is very interesting in our field because most of the measures we use to look at connectivity are measures of functional connectivity. And by their very essence, they will have within them built in a certain degree of inaccuracy. And in many cases, sometimes as much as 50%, which is quite concerning. So what you see in front of you now is the operational definition of coherence, which is in equation 1 and then later reduced down to equation 2. This is taken from our seminal publication in 2014 in Frontiers in Human Neuroscience, which we'll get to shortly, and you'll have the reference for that. And basically what it shows is that the final normalized coherence value is taken between two locations in the brain in terms of EEG activity, where we take the cross power spectral density between these two locations, or basically a correlation of their activity within a given frequency. And then that is in our numerator. Our denominator basically is taking the power spectral density of each location within that frequency and multiplying the two of those so that amplitude or power is no longer important in the equation of calculating coherence, and it is a true cross power spectral correlational statistic that gives us coherence values. Within this equation, however, it is generally recognized that there is something called the real co-spectrum and an imaginary quad spectra, with the imaginary being activity that is not of significant accuracy, with the real quote unquote being more accurate. So it is inferred within this equation that there are degrees of inaccuracy built into this model. So typically when we do EEG coherence, again, we use what are called paired coherences, or what we'll call bivariate coherences between two locations at a time. These could be on the surface of the brain, or they could be at sources within the brain, but there's still two locations where we're measuring coherence, as we've talked about in the previous definition. For a long time, this served as the standard method of establishing relations between signals, being cross-correlational and coherence in nature. That is the measurement. And in large degree, that is still the primary strategy used by most clinicians and even many researchers in the field. However, this is changing. And the reason for this is that directionality cannot be measured in any way. The accuracy of the correlational statistic may be misjudged because there may be third, fourth, and fifth influences that are not taken into consideration. So let's say, for example, we're measuring the correlation or the coherence between C4 and F4, and we measure it at a certain value, but let's say that P4 is influencing both of them simultaneously and causing the relationship between C4 and F4, but we don't know that when we just measure coherence between C4 and F4. These levels of analysis need to be incorporated, of course. It also does not consider the influence of inner electrode distance. So one of the things we know of coherence statistics and any correlational statistics of EEG activity is the closer the sensors are to each other, the higher the value of coherence. The further apart they are, the lower the value. This is not accommodated for when we look at coherence measures. And again, we cannot measure reciprocity of interactions because we can't measure directionality to begin with. Again, it's just a correlational statistic. So what we're going to do here is we're going to take a case example that we'll go through during this talk and compare measures of effective and functional connectivity so you see how they play out in real practice. So this is an example of a diffusion tensor image of, on the left side, a teenage girl with what's called a mu rhythm suppression deficit or a mirror neuron dysfunction. This is a phenomenon that occurs in autism where they have major social and motivational behavioral deficits. And on the right, you see a similar DTI image for someone without such symptoms, essentially functioning normally based on their development. There is a green ball drawn on each of them, which represents a region of interest ball that shows all of the fibers and pathways running through that region. So that region is the mirror neuron regions that has been detected by research in chimpanzees. Of course, it works different in humans, but that was our region of interest. In the individual with the mirror neuron dysfunction, with the normal development, we can see that their activity goes all the way up to the prefrontal cortex, goes through the mirror neuron location, down into the temporal and limbic system, so they can fully process social and emotional information that can be regulated by the prefrontal cortex, and they can interact as they wish. In the individual with autism, that does not occur. Their prefrontal cortex does not get activated. Their temporal and limbic system does not share communication with the frontal lobes, and the pathway goes up straight to the parietal lobes, causing sensory anomalies while they're trying to engage and interact with other people. It is very clear to see that these pathways are disrupted, And that if we could look at this in EEG technology, we would see clear disconnections between frontal and medial and posterior temporal regions of the brain. So what happens if we look at this same exact person's bivariate coherence measurement taken from their EEG with their eyes open? We see blue and red lines in delta and theta that reflect low coherence in blue and too high coherence in red, but none of these lines are showing problems connecting frontal to temporal and posterior temporal regions. So essentially, the deficiency that we know should be there is not detected at all by this analysis. And in fact, it identifies separate areas in the frontal lobes and the back occipital and parietal lobes where there are deficiencies. Now, there may be problems in these areas in autism, but that is not the problem that we're looking at in this particular example. This mirror neuron problem should be reflected in other ways, and it's not reflected at all in this paired coherence measurement. We then go to what we call multivariate coherences. These are still correlational, but they're now incorporating all 19 electrode pairings to understand our analyses. And we can see separations. Let me just draw this quickly if I can, so that you can see it. We see separations which reflect low coherence from the frontal lobes to posterior temporal regions in basically every frequency, delta, theta, alpha, and beta. And this matches much closer the DTI image and the anomalies we saw previously. So our multivariate coherences reflect the reality that we need to see to reflect the structural connectivity greater than our paired coherences do. And in fact, back in 2004, which was the first study done by this Polish EEG scientist and engineer, where they did a simulation study of comparing bivariate and multivariate connectivity approaches. They had a simulation model that you see up top, where they know that the connections go from 1 to 2, 1 to 3, 1 to 4, and 1 to 5. On the right-hand side, they did a multivariate autoregressive statistical analysis, where essentially they were able to show that using that technique, they could detect those connections precisely. On the left-hand side, they used a paired coherence technique, and they did detect those connections, which are shown as lines in black on the bottom left diagram, but they also detected connections in red that essentially did not exist. These are what are now termed spurious flows, and they show activity that is not actually present. So when we get an analysis based on paired coherence values, we may have some information that is correct, we may have some information that is not correct, and we do not know which is which in that analysis. So in fact, 4 of these connections were correct. 1, 2, 3, 4, 5, 6 were not correct. There are more that were incorrect than were correct in this specific example. To look at another example, this was another experimental manipulation where there were 5 locations of activity, and the connections were known from 1 to 2, 2 to 3, 3 to 4, 4 to 5, back from 5 to 1, and back from 5 to 4. And then these data were looked at in multiple ways of looking at coherence and similarity. So first we see auto and cross spectra that show activity basically across every connection. Coherence is shown on the bottom, where the typical coherence algorithm is on the left and imaginary coherence on the right. Again, you can see that most of these boxes are filled in, Obviously, we would ignore the diagonal, starting up in the top left to the bottom right. So now you're comparing a single location to itself. But in every other box, you see a relationship, and there shouldn't be in every other box, based on our experimental manipulation. We then look at phase differences or time delay. Again, in virtually every box, we see a relation. We then go to the cross to the top right, and now we're using multivariate autoregressive techniques. So this one is called partial coherence, which is not directed. It's still a correlational technique. And we can see that there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 boxes reflected, when really there should only be, back to our diagram, there should be 1, 2, 3, 4, 5, 6. Back down to the bottom left, distributed transfer function, which shows more. And finally, partial directed coherence, which shows our 1, 2, 3, 4, 5, 6 connections that are our true connections. So this becomes our multivariate autoregressive technique statistic that is the most accurate. It's called partial directed coherence. And when we talk about using this for our effective connectivity, this is the statistical measure that will be utilized. But other measures of correlational and coherence and phase measurements, we can see have inaccuracies built inside them. So let's take an example of effective connectivity. This is the reference I talked to you about earlier. So this is our 2014 study published in Frontiers in Human Neuroscience on a special issue devoted to understanding autism. Was entitled Using Quantitative and Analytic EEG Methods in the Understanding of Connectivity in ASD, where we compared measures of coherence, multivariate coherence, S-Loretta coherence, and Granger causality connectivity, and showed that the Granger causality was the most accurate in terms of resembling structural connectivity. You can freely download this article at frontiersin.org, which is, and all of their articles are open source, or at our website as well. If we take another example where we look at major depression, in the upper left-hand corner, we see a classical PET scan for someone with chronic depression, where yellow and red activity reflects greater perfusion of activity in the brain, and blue and green do not. So what you typically see in depression is that there's a diminished activity or hypoactivity over left frontal, temporal, limbic regions in the brain. And that is generally considered to be our understanding of what causes significant depression, or is the consequence of having significant depression. In the bottom left, you see a summary diagram of a meta-analysis of fMRI studies, fMRI connectivity studies in depression. And the blue areas you see now reflect areas that are agreed upon to be involved in depression. Again, left frontal, temporal, limbic, amygdala, other areas of dysfunction. And in the upper right, again, are regions of connectivity that are dysfunctional in depression. And you can see the greatest degree of pathway connections to be found in that same regions of the brain across left frontal, temporal, limbic areas of functioning. So if we do an analysis, we should see that those are the areas that are affected. So we do an analysis with some major depression, and on the top you see our measures of paired coherences. Again, not one of these findings shows a problem across those regions of the brain. So this analysis completely misses what we need to see in depression to treat it effectively. Row 2, you see our multivariate coherence analysis, which does in fact show very severe hypocoherences across the left temporal and frontal lobes of the brain in alpha. As a lot of us know, alpha is a hot frequency for depression, but you can see it in others as well. Here you see it in theta. And then at the bottom, we look at our independent component analysis Granger Causality metric, where our dark blue lines reflects underconnectivity. And here we see this across the same critical regions of the brain over the left temporal, frontal, and limbic regions that we expect to see in major depression. So our multivariate metrics show us the problem. Our paired coherence metrics do not. And our Granger analysis shows us more of a three-dimensional causal nature to the problem, leading to enhanced effective connectivity resembling the actual structural connectivity that we should be seeing. Okay. Okay, so from there, we're going to move on to try to understand some of these structural brain pathways. So probably the best atlas that's been created for this was published by Marco Catani a few years ago, where they actually took MRI diffusion tensor images and compiled them to show us what these structural pathways look like. And you can get his original article, which was published in, I think it's 2008. And then later they wrote a book that's an atlas of human brain connections, which I believe was published about 5 years ago. Very informative information. And for anyone working in this area where you are trying to apply connectivity training, understanding those networks is crucial. So here's just an example of some of them. These are in French, so excuse some of it. But the anterior commissure is a pathway connecting both frontal lobes. The corpus callosum, we're going to go from left to right. Most of you are probably familiar with. This is the greatest degree of white matter tracks in the brain, and it connects the left and right cerebral hemispheres. The internal capsule on the upper right-hand corner in green goes all the way from the motor cortex down through the basal ganglia into the brainstem and cerebellum, and is responsible for motor function within the brain. Middle left shows us the cingulate. We are used to talking about the anterior cingulate, which is in the posterior aspect of the frontal lobes, but the cingulate gyrus itself goes from the anterior to middle and posterior brain regions and into the limbic system itself as well, and is considered by some to be part of the limbic system. Next is the Fornix, which is a very important pathway within the limbic system, important for memory functioning. The uncinate fasciculus then importantly connects the anterior temporal and orbital frontal regions of the brain, which is very important in emotional regulation, especially in traumatic brain injury where this pathway is often severed, but it's important in other areas of functioning as well. Bottom left, you see the inferior frontal occipital fasciculus, a pathway connecting all the way from the back to the front of the brain. Then we see the inferior longitudinal fasciculus connecting occipital to temporal lobes, very important in reading and understanding in the left hemisphere and in social perception and social activity in the right hemisphere. And the last one being our arcuate fasciculus, which in the left hemisphere is considered the critical pathway for language function that connects Wernicke's and Broca's and many other language regions responsible. And those have to be fully functionally connected for full language function to be seen in an individual. This is another group of pathways that you can see again that shows association fibers up top, which are essentially the ones we've already seen, including in dark blue, the inferior frontal occipital fasciculus, In light blue, the uncinate fasciculus. In red, the superior longitudinal fasciculus. Green, the inferior longitudinal fasciculus. And in yellow, the cingulate bundles. And then we can see thalamic radiations and projection fibers below. So this gives you an introduction to what some of these pathways are. Again, I would highly recommend looking into these further so that you understand them, because when we train networks, we should be training across known network pathways. This takes us into what's called graph theory, which is a way of helping us to understand connections in the brain. And that's what we want to move into now. So graph theory is essentially a guide to help us understand these networks. And essentially, we look at what are called nodes, which are the little circles in the graph, which is basically a unit or location in the brain or group of neurons. Edges, which are the lines connecting them, which are the structural connections between these locations, and hubs, which represent regions of interconnected nodes and edges. A path, then, is the connecting edges and nodes that form a system. When we use graph theory to measure connectivity, we can measure those paths and their efficiencies, or the connectivity across any known system. In graph theory, we have elements such as hubs, which are nodes with links that exceed average, so they're happening frequently. We have low versus high degree hubs. So high degree hubs are sort of networks where there's lots of activity happening at once. Provincial hubs, where there's many more nodes and edges connected to each other. Modules, which are essentially neighborhoods of connecting features. And connecting hubs, which are hubs that through paths connect different hubs or different modules or neighborhoods in the brain. And again, this enables us to typify modules and networks and look at what we call provisional networks, the greatest one being called the Rich Club network, which we'll get into shortly, but that's the one that has the most activity occurring across those different modules and networks. And again, this helps us create a path. For example, the one you can see below, where we can actually measure these linkages and their efficiency over time. And we can do that with our connectivity metrics. From there, we take this information and we can study different pathologies related to this. So this is an example of studying default mode network activity in individuals with memory and anxiety problems. And what you can see typified here is this Rich Club network, or the default mode network, which goes from bilateral parietal to bilateral frontal regions of the brain. And these are the areas of the brain that in normal functioning should be most active when we are at rest. These help us relax, these help us pay attention, these help us regulate our behavior and remember information. When they're not functioning properly, then those things go awry. Here's another example of this in autism, where on the right, these are paths as we expect them to be in a normal individual. And on the left is a group of individuals with autism, where you see some hubs and nodes being hyper-connected, and the connections between the different nodes being dramatically stretched out or having very low connectivity. So these techniques help us to understand these pathways and systems in the brain and how they are functioning in different groups of individuals. And you can see a similar example in autism. Again, on the right, where you see many of them just bunching up together, and then different neighborhoods being dramatically stretched out, where connections going from one sort of neighborhood or module to another would be very, very problematic. And that's essentially what happens in autism. This was a study we presented on a year ago at the ISNR conference, where we measured short range and long range connectivity in developmental trauma, and found that looking at them separately, that we could detect greater problems with long range connectivity than short range in adults who had developmental trauma as children. And this helped us to conceptualize connectivity-based treatment for these individuals. Very interesting finding. So from there, we go to trying to understand some functional brain networks now. So the initial one that we look at here, again, is this rich club network, or what's called the default mode network that you see on the right in the center. So this is a network that connects both superior parietal gyri to bilateral superior frontal gyri and incorporates the tip of the caudate on both left and right sides as well. And the connections between all these locations forms what's called the rich club of the human connectome, or the default mode network. That's what it looks like. And being able to typify these networks helps us understand functioning and where we should see problems when people manifest with symptoms. So that's the importance of functional networks. On the left here, you see the default mode network, which we've talked about. There's one called the salience network, which is involved in attending and switching our focus to different things. And the central executive network, which of course is involved in attention and executive function. We've talked about the notion of functional versus effective connectivity. On the left-hand side here, you see some of these network activations typified with functional connectivity metrics, and on the right with effective connectivity metrics. And you can see that when we look at effective connectivity, the locations involved become much more focused. We see them much more clearly, and there's more noise when we're looking at functional connectivity metrics, even through fMRI. So that makes sense. That's what we should see based on the definition and what we now understand about the difference between functional and effective connectivity. So it's important to keep in mind when people talk about these functional brain networks that they are based almost entirely on metrics of functional connectivity. And as a result, we should question the preciseness of them. The general locations may have some accuracy, but the precise locations may not be perfect because of this information, because of the way they were analyzed. Again, here's our default mode network. In the middle top, that's kind of the typical image you see. And we've talked about the regions involved in the default mode network. This is our salience network that connects the anterior insula with the cingulate gyrus, being very important in, and the posterior parietal regions as well, being very, I'm sorry, not in the salience network. The salience network is the one in the middle, central executive on the right, default mode on the left, and you can see the difference between them. So the salience network tells us what's important, what we need to do, what we need to act on, what we need to focus on. The central executive network tells us how to act on those things. The default mode network tells us when not to act and when to be calmer. If we look at the salience network from an internal and external view, you can see the insula and the prefrontal cortex and the cingulate gyrus, and those are the regions involved in that network. Moving on to the central executive network, which of course is involved in things like attention, mental switching, maintaining our attention, inhibition, control, self-monitoring, problem solving, all these executive functions that we often talk about in our patients, and this is the network involved in regulating that. The frontal lobe, since the time of Alexander Luria in the in the early 1900s, is known to have large regulatory input on multiple brain regions, including other areas, the cortex, the basal ganglia, the thalamus, and even the cerebellum. And of course, these influences are reciprocal, as all brain network influences are. Don't let anyone tell you that they're not. All of them are reciprocal, and once we gain the ability to measure the reciprocal nature of these connectivities, then our interventions becomes much more effective. What you see now, we've seen a little bit before in the case of depression, is the affective network in this magenta color, and we've talked about what regions are involved in that as well. And social networks. So on the left-hand side, we see the typical social networks going, including the fusiform gyrus, posterior superior temporal sulcus, the amygdala, and orbital frontal cortex. Of course, this is very important in autism and any other conditions with major social problems. And on the right, you see the mirror neuron network that involves the parietal lobes, inferior frontal cortex, which was the mirror neuron location we talked about, and the portions of the insula as well. And both of these networks are critically involved in social and motivational functions. And lastly, to the limbic system, starting with the frontal lobes going down into the amygdala and other regions of limbic circuitry that are important, of course, in emotional regulation. And every region connected to those regions are also important in that as well, including the cerebellum that we're finding, and its connections with the amygdala and even the frontal lobes are very, very important in developmental trauma and other conditions that lead to limited emotional regulation or typical emotional regulation. So very important in understanding all of these functions. So once we understand this, can we now actually assess effective connectivity in everyday clinical practice? With the person in front of us, can we measure their EEG and get these measurements that will help us understand these systems better? Well, I hope the answer to that is yes. And that's what we'll be talking about in our last few minutes here and in part 2 next week. But if we're to do this, we need network and causal effects to be measured between neural elements. We must use statistical techniques and statistical modeling that assess causality and interaction. And this requires highly complex data processing and modeling techniques such as ICA, partial directed coherence, and Granger Causality, techniques that are not currently part of routine QEEG databases. And those are things that we can discuss. So again, we go back to our case example of our DTI data of our teenage girl with mirror neuron dysfunction, which again should show dysfunction in the right hemisphere, which is what we're looking at in a sagittal view here. A disconnection from frontal to medial and posterior temporal regions of the brain, regions that are essentially intact on the image on the right-hand side. Again, our paired coherence measure on the bottom does not detect that system at all. Our multivariate coherence metric does detect it, but it doesn't tell us causality. It doesn't tell us which regions are causing what. It doesn't tell us how they're reciprocally affecting each other, and it doesn't necessarily tell us the most critical locations that we want to know for training. Although it does tell us the system involved. And I will say it doesn't tell us the source of that activity. We then move into what's called Granger Causality, which is based on performing an ICA analysis initially and then moving to the Granger Causality analysis. And the way you interpret this map is that each of these circles or nodes is a source of brain activity that we've localized through a method called dipole localization. And we've now established causal elements or relations from each dipole to every other one. And we have a measurement. So we have a measurement from the partial directed coherence from 0 to 8. So that can go from 0 to 100. But in this teenage girl, her maximum connectivity is 8 out of 100. That tells you the degree of underconnectivity in her brain. And the lowest values on the scale are in dark blue. Those values go from about 0 to 3. So that tells you how low her connectivity is from prefrontal regions down through medial and posterior limbic sites of the brain, and even some parietal areas as well. That typifies exactly what we saw in the DTI image. Let's go back to that a little bit, if we can. What we saw here is obviously a dramatic disconnection from frontal to temporal regions, but a move up to parietal locations that are also involved in this pathology. So we should be able to typify all of that in our analysis, and that's exactly what the Granger causality gives us. And that would then tell us the exact locations and sources to train connectivity in our neurofeedback protocol. It's a very elegant way of now looking at effective connectivity, which brings us back to emulating structural connectivity. That's what makes it effective. It tells us what's really happening. Again, our example of major depression, where we should see dysfunction over the left frontal temporal limbic regions of the brain. Our paired coherence analysis on top doesn't show us that. Our multivariate coherence analysis shows us the system where there's a problem in alpha and some in theta. And again, our ICA and Granger causality show us the precise locations and sources of this dysfunction. Again, we're looking at the dark blue lines and the paths between them as showing us where the problems are in the brain. And when we train that, we've shown through research that major depression can be treated very successfully with multivariate connectivity training once you have this analysis and can understand it. And we've even shown long-term follow-up and effects of that training so that people emerge out of depression in virtually every case, in about 94% of the cases. And at 2-year follow-up, they stay improved without any repeated treatment. So a very effective way of dealing with that. So once you have this effective connectivity information and you really know what part of the brain is causing these symptoms, then you can move towards defining neurofeedback protocols that are much more effective. So that's the point at which we want to take a break in terms of our part 1 of our webinar. You can find more information about everything we've discussed at our website, where other webinars, presentations, and articles are located, including the 2014 article I referenced, and other PowerPoint presentations and other information. And we will move on to part 2 next week, where we'll talk more about actually applying this in more cases and neurofeedback applications as well. In the meantime, I'm happy to spend some extra time here today talking about any questions anyone might have. So there's a chat button on the bottom where you can go, and if you click that, you can write in a question that I'd be happy to answer while we're sitting talking about this information. So if anyone has a question at this point, feel free to just post that, and I'll just answer them until there aren't any more questions. I will say that we're happy to work and help any of you that would like to look at this information by helping you with your own EEG data and neurofeedback as well. Is part 2 of the seminar this Friday at noon? Yes, I'm pretty, you should have that information in the emails that were sent out. And I do believe that's when part 2 is taking place. Let me just check for you real quickly to make sure I know that for a fact. And yes, that is this Friday the 14th from 12 to 1 Central Standard Time. There were a couple other questions about anyone that's missed portions of the webinar. Once the total webinar is completed, we will post the webinars on our website, so you'll have access to them at that time. Alina asked a question, in terms of physical requirements for connectivity training, what's involved? So the model we use for connectivity training right now is a 4-channel training model. So you would need a 4-channel amplifier, and you would need software that has implemented this model. So currently, the software platforms that have implemented this include Eeg, which is the old EEG Spectrum software, BrainMaster, Avatar, and BrainMaster in general. And there are other platforms that are be adding this, including, I believe, Thought Technology and some others. And there are some others that have programmed this as well. So you should be able to gain access through most platforms. If not, we'd be happy. This information is freely published, and anyone can feel free to program this into their software. There's a question about working with children with fetal alcohol syndrome. Would this be a better assessment to work with these children? I definitely think that it would, and it will give you much more precise and accurate information. Is this a useful moment to speak about mapping more than 19 sites? We might save that question for part 2 of the webinar. So Ellen, if you could remember to ask that then, that'd be great. Anna asks about, do you have a reference list for the material presented on autism? I'd be happy to provide that if anyone wants to email me, and I'll try to have a full reference list for part 2 next time. And then next question, how this approach differs by just adding basic amplitude training from a more simple QEEG analysis. So connectivity training and amplitude training are not the same, and you cannot get the level of impact based on amplitude training that you can from this type of training. And in part 2, when we talk about some of the research behind our model, I'll talk about that very issue specifically. And you'll see some of the data about that. Do you have any good layperson references we can give to clients to help them with multivariate coherence training they're doing? Wow, that's a very interesting question. I do not have information like that. I think that would be very useful. And I think it would be very useful to have even sort of short video presentations about that. On our website, we do have some interviews of some of our practitioners that use this model. And I think that can be helpful to introduce to clients. And we are hopefully going to create sort of a short video that explains this approach that can be used for professionals and clients alike as well. That was a very good point. Any other questions at this point? Okay, so at this point, I'll thank everyone for attending and look forward to meeting again on Friday and answering any questions you have at that time. Well, you all have a very good day. All right, good afternoon, everyone. Everyone, including Mark Pesner. Hi there, Mark. So we're gonna get started here because we've got a lot to cover today. So this is part 2 of the webinar on understanding brain networks. Let me pull up our slides for today and we'll get started. So this is part 2. Okay. So again, just to review, these webinars are made available and they include access to BCIA and APA CEU credits. The information that we're going to provide is as accurate as can be, is based on empirical scientific data and information, and hopefully you all will benefit from it. I am one of the directors of Integrated Neuroscience Services that provides QEEG and neurofeedback consultation services to people in the field. Just need to declare that as we talk about the remainder of this information. Okay, so what we're going to start, there's lots to get through today, and I'm hoping we can do that in an hour and then have time for questions at the end. If you do have questions, you can type them in the chat function, and hopefully at the end, we'll be able to spend an extra 5 or 10 minutes going through those. What we're going to start with is a series of case studies that look at, in everyday QEEG activity, looking at effective versus functional connectivity measures. These are clinical cases seen in our everyday practice where we perform normal QEEG procedures that, of course, include bivariate or pairwise coherence analyses. They also have included multivariate coherence analyses utilizing the NeuroRep software, which is based on a principal components analysis. And then in all of these cases, we've also applied independent components analysis and Granger Causality analyses, which, as we've talked about, is our measure of effective connectivity. So before we do that, we've got to talk a little bit about what this technology does and how it works. Independent components analysis is a technique used by the preeminent neuroscientists in the world. It's been applied to fMRI, MRI data, EEG, MEG data, and it's been used in the following institutions, and the following journals have published information, including such analyses. these are all kind of heavy hitters, and the people involved in this are really tops in their fields. The original publication was put out by Scott McCaig in 1996, called Independent Components Analysis of EEG Data. And what you see on the left-hand side is a brief EEG channel tracing of 1, 2, 3, 4, 5, 6, 7, 8, 12 EEG sensors. ICA, which is a statistical procedure that at its heart basically reshuffles the deck and puts statistically unique information into different categories or different components. And those are labeled from 1 to 14. So you can see it changes what's at the channel level and puts it in the component level. So for example, component 14 is all made of muscle activity from several different sensor locations. It's not just one. Component number 1 is probably made up of theta or alpha activity from several different sensors, not just one. So it reshuffles the deck and puts them into unique categories where we can study and utilize them for other analyses. This is a later publication of what's called EEG Lab, which is an open source toolbox to use in performing ICA analysis. This is what we use in all of our analyses. And Scott McCaig and Arnaud Delorme were the primary engineers of this, and people have helped ever since that time. And essentially, again, it takes the scalp EEG and converts it into what are called sources, which are the components themselves. In EEG lab terminology, they're called source activations. So they are the components and the EEG tracings of those components. Some of the components are then made up of artifact, and some of them are real EEG. And what happens in an ICA analysis is you essentially delete the ones you think are artifact, and you keep the ones you think are real EEG data. And that de-artifacts all of your EEG data in a very pristine fashion. You can then, at the bottom right here, get what are called scalp projections, which is basically a topographical map of the source activations for each of the components. And you can look and study those as well. Here's another example where you can very easily see a component of an eye blink or lateral eye movement in this case. This fourth one is an eye blink. 12 and 15 and 19 are obviously EMG activity, muscle activity, which you see around the temporal bones, which are pretty obvious. And then there's others in here that are real EEG activity. So it enables us to decide and choose what we want to keep and what we don't want to keep. The pipeline of our EEG processing system includes the import of the EEG data itself, the independent component analysis, a process called dipole source localization, which takes each component and finds its source. That is then converted into what's called multivariate Granger Causality, which is the connectivity analysis, and followed by graph theory metrics, which enable us to look at the entire network and its functioning. Granger Causality is essentially, it's not a new concept. The mathematics were developed by Granger in 1969. Its application to EEG in terms of studying connectivity was probably started in about 2005. So we're a little more than 10 years into it, and it's certainly been refined since that time. And essentially what it does is it infers causality from one source to another. So it's sort of like a multiple regression equation where you can determine how much one variable causes another. You can also do it in the opposite direction. So you can determine the directionality and measure reciprocal causality. And as the equation applies, you can do this from any number of sources. This could be 3, 6, 64, 128. That's irrelevant. Granger causality can process any number within the equation. So this helps us to do our connectivity analyses and derive all of this effective connectivity information. And it is not just correlational, it shows causality, directionality, and reciprocal causality, as you'll see. So here's an example of an EEG tracing on the top before it's artifacted. The ICA procedure is applied to this data, and you'll notice that this is more than 19 channels. So this is 32 channels, actually 30 channels of EEG data. And we'll talk as we go through this that there are certain advantages in this approach to having more than 19 channels of data, although you can do these analyses very effectively with 19 channels as well. Once the ICA is performed, you can see the bottom is what's left over. So you can see that all of the artifact has been removed and the EEG is very even and settled, and we can see ebbs and flows in actual EEG activity, and the eye blinks and other movements and muscle activity have all been removed. From there, we get our source activations and our component properties. So up top, you now see a two-dimensional map of each component and its activity. Typically, the way this works is red is a positive direction, blue is negative in the EEG. So you can see where the activity is coming from. And then below that, we can actually plot a three-dimensional activation as well, so we can see where it falls on a three-dimensional scalp head in addition. That information is then taken and we can plot what's called a component spectra, where we look at power spectral density in different frequencies, and then we can measure each component and how much each one contributes to the overall activity at different frequencies. Very informative information. And then we can look at, sorry for the graphics on this, but each component produces several statistics that enables us to determine if in fact it is real EEG data. So we get a spectral plot, we get the EEG tracing itself, we get the topographical mapping, we get what's called a roster plot, which shows us ebb and flow in the EEG, and we get the dipole mapping, which shows us if it's actually in brain structure or surface as well. So all of this is used to determine good data versus bad data, and of course the bad data is then removed. From that, we then get our dipole localizations. In this particular case, there were 8 sources of activity. This shows the dipoles in a traditional MRI array of a horizontal, sagittal, and coronal view. And you can then determine where the dipoles are, where the sources are coming from. And in fact, we can use what's called a Talairach atlas to determine the exact location of these, including Brodmann locations, but even more specific locations within both cortical as well as subcortical brain structures. The ability to localize to subcortical, limbic, and other brain regions, and to have greater specificity becomes dramatically enhanced the more data you have in your EEG. And data is added by adding more channels, by having a longer recording, and by the sampling rate you use in your recordings. So if any of you want to capitalize on this and you record at 128 sampling rate, that should be increased. And in many modern amplifiers, you can record at least 256, and on some you can go as high as 1,000. The Mitzar amplifiers are at about 500, so that adds to the purity of the data as well. This is now a plot of what we see in Granger causality, and it shows us connectivity over time. Each one of these sequences is traditionally about a minute apart from each other, and we can see how the ebb and flow of the connectivity evolves and changes. Again, the clarity of this is not very clear, but typically the dark blue lines represent a pattern of underconnectivity, and the red or orange a pattern of higher connectivity. And we can interpret how it changes over time and the level of it for each individual patient. This is our measure of effective connectivity. And back to our previous webinar, that this uses the metric of partial directed coherence for those of you that are interested in knowing that information. This is a second plot we've added probably about a year ago or more, and this is what we refer to as reciprocal causality. So what it does is it takes the activity from the Granger Causality plot, and it measures the cause, say, from example, from 1 to 2. We get a specific numerical value, say it's 0.2, and we get a value going from 2 to 1. Let's say it's 0.1, and we subtract them. So there'll be a difference of 0.1 in the favor of 1 going towards 2. So the arrow will then point towards 2, telling us that the causative value from 1 to 2 is higher than 2 to 1, which means that 2 is now a weaker link in this chain of connectivity. And we can now judge those balances and help to understand which regions of the brain are most critical in understanding this patient's functioning. So we've got a couple arrows pointing to the back right and some to the frontal lobes, telling us that their balance of problems is in multiple regions in both of these locations, and it's the connections between them that are very crucial. In some cases, it's more one location or another, and we might emphasize those in our training. But all of this information is very helpful and informative, and goes far beyond what you can get from correlational statistics. Now we know directionality and reciprocal directionality. Okay, so let's look at a few cases here. The first one is a case of autism. This is a 15-year-old girl who presented with symptoms of repetitive behaviors, flapping, spinning, trouble with transitions, temper tantrums, social impairments, avoid crowds, a lot of anxiety, was on multiple medications. She's a 10th grade student, doing very poorly in school, especially in reading and mathematics. And our assessment revealed impairments in attention, social perception, mathematics, and great difficulty in social perception and matching someone's affect or the emotion they're expressing with their true emotion. And that impacts quite significantly her social skills. Okay. Our assessment, again, was most likely a 32-channel EEG. And again, we can see in the back here, most of her components are actually in the back of the brain. There's 9 components total. 8 out of the 9 are in the back, only 1 over the frontal lobes. A lot of people tend to think of autism as a frontal lobe disorder, when in fact, it tends to be a processing disorder over occipital, parietal, temporal regions of the brain. And we can see here in her dipoles that the majority of them are over occipital, parietal, and right parietal regions of the brain, with more of them over the right hemisphere. The right hemisphere specializes in social perception and social skills and social anxiety, which makes a lot of sense in a case such as this. We go from there to her Granger Causality, which now shows a pattern of underconnectivity, the dark blue lines, going from occipital, middle parietal to right parietal regions of the brain. And those would then be areas we'd want to train in our neurofeedback protocol to help her improve. This is her typical QEEG mapping that did in fact show some beta over the back of the brain and the right parietal temporal area as well. Her S. Loretta, interestingly, was more on the left side, but also in the parietal region. And there was another S. Loretta that was actually in the right parietal location, which matches much closer to these dipoles. So if you have any concerns about the localization, S. Loretta and dipole localization almost always match. But the dipoles give us more detailed information of multiple specific locations and enable us to measure connectivity across those locations. Here's our coherence measurement. And here, what's missing? Well, what's missing is problems over the area we just saw in our effective connectivity where they exist. We can see that in the multivariate coherence plot, but not in the paired coherence plot. So again, as we talked about last time, this paired coherence metric actually misses the point. We would actually train on the left when in fact we should be training on the right. Again, there's the Granger causality to compare to that. So you can see how in this case, this information gives us much more precise and much more effective, accurate information. Case 2 is a 7-year-old boy with ASD and epilepsy. He has a chromosomal syndrome called Lennox-Gastaut syndrome, which is well known to be essentially untreatable. It's hard to diminish seizures in such a case. He has history of seizures and they're daily events, actually more frequent than that. They've tried multiple medications, multiple supplements, and he has very, very significant problems in everyday functioning. This is a tracing of his EEG activity, and you can see that his seizures are almost occurring all the time, honestly. There are EEG tracings where they don't exist, which is good. They're not life-threatening, but he is going in and out of seizure activity almost constantly throughout the day. The red bars you see up here are likely seizure events across his entire recording, and you can see they're occurring very, very, very frequently. And spike events, which are these, occur, of course, even more frequently, like we saw in the actual EEG tracing. This was his assessment. This was based on a 19-channel EEG assessment from another clinic that was sent to us for analysis. And we can see in the components, activities over the frontal lobes and the left frontal regions especially. He has extremely impoverished speech that has been affected by his seizures. And we can see 4 of the 5 components are all over the left hemisphere with a special focus over the left frontal region. And his Granger causality shows a pattern of low connectivity across those regions with occasional burst of hypersynchronicity, which is what happens when you have a seizure. That goes to an entirely hyper-connected state, and at other times it's actually too hypo-connected. So our philosophy is to train up coherence so that his brain can prevent that from happening, which has shown promise in treating seizure disorders. This was his Loretta, showing very significant localization of middle frontal and orbital frontal floor pathology into the limbic system. And his coherence mapping is here. In the multivariate neurorep mapping, we do see a frontal lobe disconnection. And we even see that in the paired coherences, but it's actually mixed with the hypercoherence. So it's kind of unclear what you would want to train in such a case, whereas with our other Granger causality information, it becomes more obvious. Case 3 is a case of developmental trauma, an 18-year-old woman seen who suffered verbal and physical abuse as a child and as an adult as well, has also had a rape experience as an adult and has had significant problems with very severe depression and anxiety as a result. This is her trauma symptom inventory, which is a psychological scale we use to measure some of these difficulties. And any T-score above a 65 is significant. So you can see very high levels of anxiety, hyperarousal, depression, intrusive experiences, dissociative events, sexual disturbances, all sorts of challenges that she faces. This was her routine QEEG, where we could very clearly see right parietal temporal dysfunction, not surprising in a case of trauma, and some on the left as well. The Loretta imaging localizes to the right middle temporal gyrus, as well as to the left middle frontal gyrus. Our components, again, based on 19 channels, is in these different areas we've seen. And here are the components across different areas. Left frontal white matter, right middle precuneus, and right parietal temporal frontal regions, which we know are all implicated in trauma-related cases. Here's our coherence measurement, which again, the paired coherences kind of misses the boat. We can see some of the right hemisphere low coherence in the multivariate coherence, and the Granger causality is much more precise in showing us where the greater difficulties are and between what locations, and how they may change and evolve over time. Here we see better functioning in the left hemisphere than in the right hemisphere. So in such a case, we might choose to train on the right first. And in fact, if we look at our arrows here, we can see multiple arrows pointing to the right hemisphere and some to the left frontal and precuneus as well. So all are involved, but the right-sided one has 1, 2, 3, 4, 5 arrows, and the other side's less than that. So that gives us more information about what to target initially. Okay, so that takes us into the treatment itself. So about 6 years ago, we created a type of neurofeedback we call 4-channel multivariate coherence training, where we take the type of information in terms of assessment we've been talking about, apply it to neurofeedback. The primary article that validated this approach based on a sample of 174 subjects was just recently published this year, 2 months ago actually, in Frontiers in Human Neuroscience, which is an open source journal with an impact factor of 3.8, which is very large. Most neurofeedback journals, for example, have an impact factor of 1 or 1.2. So this goes out to more more people in neuroscience, even beyond those that just do neurofeedback every day. And this is freely available for you to download from this source or from our website. This says that this article has been read by more than 1,300 people worldwide in the 2 months it's been published. And you actually get to see those numbers grow over time. It's pretty cool where you get to see that from their website. So essentially, what is it? What we do is we use 4 channels of EEG. Those are selected based on your EEG analysis, as we've talked about. And we calculate multivariate coherence between and across all these channels. So for example, we calculate coherence from A to B, A to C, and A to D, following these black lines. And we take the average of those 3 values, and that becomes the shared coherence value at point A. We do the same thing for B. Here B is actually, I think, these green lines. The color is wrong on that scale there. So we take the coherence from B to A, B to C, and B to D. We average the 3. and that's our value at point B. C, which is in blue, we take the coherence from C to D, C to B, C to A, average them, and for D, the same thing, and we average that. So we get a shared coherence value, which goes from 0 to 1. In most softwares, it's multiplied by a factor of 10, so it's a larger number we can work with in training. In Eeg, it is multiplied by a factor of 10. So we can see the values at each of these locations as you're training. And then we calculate one metric called QPS average, which is the average of the values at A, B, C, and D that's calculated over time, over and over again. Okay? And that becomes our reward band. So we can decide to train up coherence across all connections in whatever frequency we wish, at whatever locations we wish, based on this one metric, or we can train it down if we want to decrease hyper coherence or hyper connectivity. At the same time, we use 3 amplitude inhibits based on if in our EEG analysis there's too much theta, too much alpha, too much beta, or EMG activity, we make those choices. So our protocols are always a combination of our reward band, where we're training multivariate coherence, and amplitude bands where we're training down amplitude, very typical inhibits. But now it's calculated an average over 4 different locations simultaneously. And that's how it works. So the primary mechanism is what's called QPS average. This is the statistical calculation that basically averages the shared coherences at each of the 4 sites. And we've developed 3 other modes as well. QPS deviation measures the difference between the 4 sites. So let's say site A had an average coherence of 5, but site D had an average coherence of 2. The difference would be 3. So now we're training that difference to go up or down, to make them more similar or more different from each other. QPS mod is the most recent incarnation of this. And essentially what we do there is someone gets the reward if they change the average and the range simultaneously. So let's say we wanted to enhance coherence and enhance connectivity. We would require them to increase their overall value across all 4 locations and to decrease the difference between the locations. So we'd be making them more similar, and at the same time, we'd be raising the coherence values simultaneously. We think this may be more powerful, but we don't know. We're still studying the impact of that. But in Eeg, you can choose among these 3 options. In BrainMaster Avatar, for example, they've only programmed in QPS average, so that is your only option there. So since we created this type of treatment, we've done empirical studies on several different populations you see listed here. This includes a total N of about 600 in our multiple samples. And in every instance, we've shown this 4-channel multivariate training to be successful in reducing symptoms, in changing EEG values, and changing coherence values, and having greater efficacy than 2-channel training or other types of neurofeedback, including single-channel or bipolar training as well. So that's cut across a general neuropsych sample, seizure disorders, traumatic brain injury, autism, learning disability, depression, developmental trauma. I believe those are all the projects we've done to date, and we continue to work on others as well. So in our initial sample, which had 174 subjects of various neurological and psychological diagnoses, what we did is we mixed the group, so they were assigned to either get 2-channel coherence training or 4-channel multivariate coherence training. And what we showed is that when we looked at change in power over about 15 sessions, actually 15 sessions with 4-channel and 20 sessions with 2-channel, the 4-channel group doubled the amount of change in power than the 2-channel group did. So it was twice as effective in only about 75% of the number of sessions. And when we looked at changes in coherence, the value quadrupled in those number of sessions. So if you want to change coherence, you could do it with 2-channel coherence training. But if you use the 4-channel multivariate coherence training, you get a 400% increase in the amount of coherence change. We've seen positive results in seizure disorders, where your typical SMR training gets about a 50% reduction in seizures, and the 4-channel training in this study had an 81% reduction across, again, about 15 sessions. In traumatic brain injury, we've seen neuropsychological deficits improve in multiple domains to almost normal levels, and we've seen symptoms reduce to almost normal levels, no matter how long it's been since the traumatic brain injury. In a study of autism, we've seen outcomes improve from about 81% of the sample improving with 2-channel coherence training to 98% with 4-channel coherence training. And we see a greater number of people changing their coherence values and reducing their symptoms by a greater amount. In a study of mu suppression problems, which causes social deficits in autism, we've seen individuals that the 4-channel training has a greater impact on coherence and a greater reduction in mu-related difficulties. So they're now able to suppress mu and their social skills improve. And one of our more well-known studies was with learning disabilities and dyslexia, where we showed a 1.6 grade level improvement in achievement scores in just 15 sessions. We also showed improvements in depression that far outweigh those of psychotherapy and a waitlist control group. And at 2-year follow-up, these individuals maintained their level of improvement, which you can see here. So in multiple regions, sorry to go through so quickly, but in multiple samples, we've seen dramatic improvements with this type of intervention. We recently, 2 months ago at this year's ISNR, we presented results of a study where we looked at does designing neurofeedback protocols based on effective connectivity measures change the outcome compared to using other measures of coherence and QEEG data. So we've talked about sort of the clarity and the specificity and the more accurate degree of connectivity measured using ICA and Granger Causality, but does it actually make a difference in your therapeutic outcome in these types of protocols? So that's what we studied. We had 45 subjects altogether. We randomly assigned them to 15 of them in an effective connectivity group, 15 in a functional connectivity, where we use multivariate coherence measures to design their neurofeedback protocol. And we did that in 2 different groups. In groups 1 and 2, they were the same patients. So they had the functional connectivity treatment first, and then even after that, we had them do treatment based on effective connectivity measures. So it was actually a stiffer test because they've already had some treatment. So what we then did is we compared clinical ratings from the patient and therapist ratings and EEG metrics to determine what their overall outcome was. When we looked at client-rated or parent-rated improvement, the effective connectivity group showed a significant difference compared to the other 2 groups. So they improved to a greater degree they were rated as better than those that use the functional connectivity measures. And you can see not only are they better on the average, but a greater percentage of them had a higher outcome than the other two groups did. So here your average ratings are about a 5 or a 6. Here your average is probably closer to a 10. You can see that in the bar plot here. But here you also have almost half your sample that has a 9 or higher, some of them going as high as 18 out of 20. You see the same thing happening in the therapist rating. Highly significant difference at 0.006, and a greater proportion of them showing higher outcome than the functional connectivity groups. And the functional connectivity groups did not differ from each other, which makes sense because they basically had similar design protocols, but it's a replication. And we've shown that effective connectivity group was superior to both functional connectivity groups. The ones that were neurofeedback naive and the ones that weren't. In other words, they already had neurofeedback and then did effective connectivity neurofeedback. When we looked at graph theory metrics of connectivity, we showed significant differences in overall efficiency, what's called path length, radius, and diameter. Those are all measures of network dynamics and connectivity, and specifically showed that global efficiency, which is the overall efficiency of the entire network, was improved to a greater degree using the effective connectivity model. And again, a greater number of them had a greater outcome. So look at this. This is really interesting. Using our functional connectivity model, we show that many of our subjects improve their coherence, but there are a small number of the sample where their coherence actually goes down after the intervention. In our effective connectivity group, that does not happen. So it almost, there's no guarantee, but almost guarantees we're moving in the right direction, and as a result, guarantees that they have better outcomes. So does it matter? Yes, it seems to matter for those individuals. Now I want to lastly take you through some case examples of how this is used on individuals, from the assessment all the way through to the neurofeedback, how it gets played out, and then how we measure progress in the neurofeedback as well. So this is an individual that presented with major depression. This is their ICA analysis up top, where we can see the source activations in different locations. And we can see that in 1, 2, 3, at least 4, if not 5 of these sources or components, there is the heightened activity over the left hemisphere. These are the dipole localizations, and you can see them over posterior, temporal, parietal, frontal, even limbic locations. And this is our Granger Causality, which now shows connectivity from 0 to 19. Let me point that out to you because that's important. So this scale here shows you the value of the connectivity metric, the partial directed coherence metric. The norm for an adult is about 25 to 40. In this individual, it was 0 to 19. The dark blue lines are going to be less than 8. So what does that mean? That means their connectivity in the dark blue connections are going to be probably only about 25 to 30% of what they actually should be. And we can see the greatest portions of those values between these areas in the left hemisphere between posterior frontal, temporal, and limbic regions of the brain. We know from our other conversations that that's what we anticipate in major depression. So we can isolate those locations, train for enhanced coherence, because this is low effective connectivity, and hopefully see that patient improve in their depression lessen. That's the idea of how the assessment is utilized. So we then go to their neurofeedback protocol designed based on that. We did a form of QPS average, which we've talked about, where we trained T5, T3, C3, and F7. So all across that network we just identified. We located their coherence band to train coherence up in the reward band from 5 to 14 hertz. If we go back, we can actually isolate the frequency in that connectivity analysis of what the reward band should be. Hopefully you'll see that in our other analyses. And then from our other QEEG frequency-based information, we get our inhibits. So we've got a theta, a low beta inhibit, and an EMG inhibit. This particular person did only 10 sessions. These sessions last about 15 to 20 minutes in duration. The result for them is shown here. Their initial Beck depression inventory started at a 28 and went down to a 4. For those of you that are familiar with this scale, this is an unheard of improvement, quite dramatic, where someone goes from severe depression to essentially very limited symptoms of depression in 10 sessions. And their anxiety reduced in a similar fashion. So this intervention was quite effective for that person, and it only required 10 sessions. Now, for that individual, we wouldn't want to stop there. Generally speaking, you need to do probably at least 20 to 25 sessions for it to stick. But you can certainly monitor that over time. That's not true of everyone. One of the complaints we sometimes get from clinicians is this works too quickly and people leave too soon. But you know, it's a process that we work through. And if we're making people better faster, we can help more people. So this is another woman, a 46-year-old woman with PTSD related to developmental and adult trauma. She had treatment for a long time, but she still had dissociative elements, reduced self-awareness, some hyperarousal and anxiety. Her EEG assessment showed the following. Again, this was based on a 19-channel EEG, where we could see dipoles in the right and left hemisphere. And this is her connectivity plot that shows connectivity issues across multiple locations. She actually had in her left hemisphere surges of sort of hypersynchronization, like we saw in the seizure disorder, where in terms of emotional symptoms, people get stuck very often. So when you see this in someone, they will have a tendency to get stuck in their depression or anger or dissociative symptoms that often happen with left frontal dysfunction. So we viewed that as a very prominent symptom for her and wanted to treat her over that brain region. So her protocol was the following, where we trained over the left hemisphere between those locations, QPS average again. So this is a sort of a protocol sheet for anybody that works with us that we share with you once we've developed the protocol so that you can implement that in your own office and in your own treatment plan. And we rewarded for increases in coherence between 8 and 22 hertz. And in fact, that was the frequency of the connectivity plot. It's a little hard to see there, but that's basically what it was. So it's easier for us to tell that. And then the inhibits, of course, you had excess alpha, low beta, and then higher beta, which is pretty common in people with trauma-related disorders. So she trained with this protocol. Sessions were about 12 to 15 minutes long. She did about 15 sessions. This was her initial Granger Causality connectivity. And then after those number of sessions, this is what was left. It's really fascinating to see that the dipoles that were in the left hemisphere that we treated are no longer there, that we're no longer detecting those problems at this point in time. But we still see some of the issues in the right hemisphere. And of course, we would continue to treat those in future rounds of training. But just in this first round of training, we see her Beck anxiety scale go down, her defensive avoidance go down, her dissociations, her impaired self-reference, her impaired awareness go down. So she became more aware, she spent less time dissociating, and her anxiety reduced as a result. What we've heard from multiple clinicians is once you do this for someone, they're also a better psychotherapy candidate, and their therapy progresses much faster as well. And doing this in conjunction with counseling is extremely beneficial. Our next individual is someone with autism, 20 years old, more with an Asperger's type of syndrome, trouble understanding jokes, symbolism, is easily distracted, very concrete, literal thinking, anxiety, trouble with change, social difficulties, not interested in social involvement, and also had a history of possible sexual abuse and neglect. So very significant problems, deficits in social perception and understanding, and all of the difficulties you might see in someone with that history. This person's assessment was actually based on a 64-channel EEG, and you'll notice how much more specific and detailed and internal these dipoles are. That's what happens when you go from 19 channels to more than 19 channels. And I know I've spoken to some of you about the utility of doing this. Again, it's not to say that it's not valid with 19 channels, it certainly is. And we've shown it's more effective than other types of coherence training or other functional connectivity models. But if you ever have the opportunity to move towards a higher number of electrodes, it's even more effective and precise in those circumstances. So here we see a pretty clear, again, localization over the right occipital, parietal, temporal, frontal regions of the brain that become clear in the dipole model. And in the Granger causality, you just see a pattern of underconnectivity between all of these locations, with the densest connections across these areas here. So that's where we did the training. As you can see, QPS average, rewarding 7 to 24 hertz, increasing coherence. We inhibited some theta and alpha, some higher beta in 2 different frequency bands, and did their training over time. Again, just about 12 sessions, and they showed dramatic improvements in social perception, on tests of social perception, decreases in attention problems, in emotional difficulties, in their sense of inadequacy, and in their attention skills, and in their social skills. So we see the desired outcome in a very quick period of time. We also have developed the ability to measure progress not only from questionnaires and psychological assessments, but from the EEG itself. So this is our last example to show you of that. A 52-year-old woman with concerns about memory and depression. Also had frequent migraines. You can see her depression was severe with a Beck depression inventory, a T-score of 98, very, very high. features of anxiety, depression, major depression, even suicidal ideation. This is their routine QEEG that showed S-Loretta localization over the left parietal and frontal lobes, and in her 19-channel reciprocal causality from the Granger Causality. Again, you can see the majority of these electrodes over the left hemisphere there that agrees with the S-Loretta, but we see more precisely what's happening here. And again, we see multiple arrows pointing to the left temporal region and single arrows to frontal lobes all over the left side as well, where there are no arrows pointing to the right hemisphere. Now, that doesn't mean the posterior right hemisphere isn't important. It probably is for her social anxiety and trauma, and the sort of Asperger's related issues. But all of the depression and trauma and dissociative other symptoms are all in here, and those are the highest priority based on where the causality shows us coming. Okay, hopefully that all makes sense. So this becomes our protocol where we map our locations to those regions of the brain, reward coherence up from 3 to 17. We inhibit theta, low beta, EMG activity over a number of sessions like we've talked about, just about 12 to 15 sessions. And her progress in that period of time, Beck depression score goes from severe to normal. So basically her depression is gone. Her suicidal ideation has lessened. Her attachment issues have lessened. Her dissociative or tension reduction or self-harm behaviors have lessened dramatically. All in about 12 sessions. Each session is only about 10 to 15 minutes long. We can also look at our graph theory metrics of connectivity and measure, was there an actual change in her EEG that we can correlate with her symptom change. So here's her path length, which is the average level of connection in the entire network. So the way you interpret these is the lower the value, the more efficient the network processing is. So her path length went from a 70 to a 30. So in those 12 sessions, she had a reduction of about 60%, or an improvement of 60% in overall connectivity. Radius is short range connections. So those frontal lobe connections right next to each other, it went from an 80 to a 34. Diameter is the long range connections. The ones all the way from the posterior temporal region to the frontal region went from 141 to 67. Dramatic improvements. So we can actually say to a patient, okay, we did this type of neurofeedback. We train your coherence and connectivity. We can show you that your connectivity changed. We can show you that your symptoms changed, and everything is related to each other. We know the neurofeedback made this happen. And we can even measure changes in power, where you see all across the frequency. Recording 1 is the blue line, recording 2 is the green line, and it actually shows things decreasing in power as well. We've recently developed more efficient ways of doing this, even more so, and are advancing this technology and our programming as we move forward in our growth and development in these approaches. So hopefully, as you can see, we covered a lot of information. I actually did get through it all. I'm pretty happy about that. But as you can see, this type of treatment, if you decide to pursue it or if you currently utilize it, hopefully you know it is more potent in some ways than other types of neurofeedback. And that has pros and cons. We have the ability to help many of our patients more so. So we can help our patients improve further, improve faster, and we can help a greater percentage of clients to improve as well. The cons are, for some people, it works faster and the changes are intense. And so as clinicians, we have to figure out how to manage those changes over time. And we really want to be careful of any overtraining, which is why we train in smaller segments. So only about 10 to 15 minutes instead of more typical neurofeedback trainings that are sometimes twice as long or more. And we do a shorter number of sessions, maybe 12 to 15 sessions before we do our reevaluations. If you pursue it, we also highly recommend these types of assessments where we are happy to help you with EEG data that you can collect, that you can send to us, and we can help analyze for you and mentor you through the process of learning how to do this. And I also think informed consent is very important for us to communicate with our clients about this approach, that it's a relatively new approach, and what they're participating in, as we should do with all types of treatment. So at this point, I'll just say thank you again for all of you for attending. Again, if you want more information, our website is integrated-neuro.com. You can call us by phone. Susan Sanders is our administrator. She can help you set up any appointments or answer any questions or access to any articles you might want from our website. And at this point, I'll take any questions that anyone might have. And I'll just stay on for about 10 minutes so we can get to as many questions as possible. So the first question is this type of training possible on the Nexus 32 with BioTrace? So the first thing to consider is you can collect a QEEG on the Nexus 32, and we can help with the analysis of that and this type of effective connectivity, absolutely. If you can, I assume you can already do coherence training, at least 2 channels with the BioTrace, so we could help you with that. It was my understanding that Nexus was going to program this to do the 4-channel multivariate training in their software. I don't know where they are in that process at this point, so you might want to call and ask them about that, but I believe it was one of their goals to set that up. Are the regions, let's see, are the regions of interest where the most power anomalies are? Interesting question. So typically the dipoles are our sources of localized activity. Those are where we're measuring the connectivity anomalies between, and those are also the sources of where the most dysfunctional activity in the brain are as well. And as a result, the power anomalies are more likely in those regions also. So I would say yes to that question. But of course, they're not just in those locations. They're spread across locations, as all EEG activity is. Any other questions anyone has? All right, well, for any of you that have attended, oh, there's another one. There seems to be more mixed hypo and hyper coherence with NeuroRep. Mark, that's a very astute observation. And I would agree with you that when we move to the Granger Causality model, what we see is less hypercoherence in general. But I want to qualify that a little bit. Since we can look at the connectivity patterns over time, so we look at it over every 45 to 60 seconds, we see these people like we've talked about that have bursts of hyperconnectivity, and at other times they have hypoconnectivity. Previously, in all other types of QEEG processing, whether it's paired coherences or NeuroRep, which is multivariate coherences, all of that is averaged. So if you have some values at 20 and some at 80, your value in between is going to be mixed. So it'll show you more hypercoherence. But maybe that's not the case. Maybe it only happens at one point in time. So this model enables us to break that apart and see that in greater detail. And as a result, we see the effect of hypoconnectivity, I think, is more crucial. Whereas in the past, 10 years ago, we talked about more hypercoherences. So that's how that has evolved. One question was, could you specify the areas with developmental trauma? So that's a very good question. And in fact, not too long ago, I think about maybe 2 months ago, we did a webinar like this specifically on developmental trauma. So you can go to our website and you can look at that webinar, and you can even get BCIA and APA credit for that webinar as well. To summarize specifically, and this is a very minimalistic description because such brief time, typical areas include the precuneus and the parietal lobes, the parietal and temporal lobes over the right hemisphere, and the frontal and temporal lobes over the left hemisphere, and the anterior cingulate gyrus over the frontal lobes. Now, there are more regions than that, of course, in individual cases, but that's probably a good summary. But the webinar and even the PowerPoint presentations on our website will give you much more detailed information about that. So you're welcome to look at those. Okay. Well, I think we're about out of time now. So again, I thank you all for attending, and hopefully look forward to any future webinars that we can present for you all. Thanks very much.